do we have our next, I think we have a pair of speakers now, Cris Salvi and Ariana Huebner. So it's a great pleasure, of course, to introduce both of them. Cris is a PhD student at Oxford and also at the Alan Turing Institute, I think. And Ariana is from TRACERx. And the title of their talk, Inference on Trees of Genetic Mutations from Lung Cancer. So over to you. I don't know who's speaking first, whether it's Cris or Ariana.
I'm going to go first.
Okay.
Yeah. So good afternoon, everybody. So Cris and I are going to be talking about a collaboration that we've started quite recently. And we're using phylogenetic trees that detail the evolutionary history of lung cancers. And currently the main aim is to try and use these trees to predict patient outcome. And so I'm going to start with some background to motivate the project and also to explain the input data that we're using. I just need to actually get the screen to work. There we go. Okay, so why do we want to look at lung cancer? So lung cancer is the leading cause of cancer-related deaths worldwide, with about 1.8 million deaths each year. And so although the incidence is quite similar to that of breast cancer, the mortality rate is disproportionately higher. And despite the fact that there is surgery and adjuvant chemotherapy and other more personalized treatments, the overall 5-year survival is less than 10%. And even if we look at the different stages, so the stage of cancers will say something about the size of the tumor diagnosis and how aggressive the tumor is, essentially. So even if we look at the very small tumors at detection, at surgery, that tend to not be classified as aggressive, that don't receive any adjuvant chemotherapy because they seem, or they're said to be healed with surgery only, we do still see that after 5 years, 40% of patients do succumb to their disease. And so there is still an unmet need to determine better biomarkers and to be able to predict outcome. And so therefore to be able to split basically these patients specifically in stage 1, early stage disease, into those that truly do not need any therapy and those that do. And also looking at these later stage cases where in some cases patients still do not develop a recurrence of their disease and therefore might not need any additional treatment, which can be harmful as well. So one important concept when studying tumors is heterogeneity. So what is well known is that there is intertumor heterogeneity. So looking at different patients, looking at different tumor types, breast cancers compared to lung cancers behave very differently, therefore need different treatment strategies. And this has been quite well known and well understood for quite some time now. Slightly newer development is what we refer to as intratumor heterogeneity, which details the heterogeneity within a single patient's tumor and looks at the different cell populations that inhabit that tumor. So previously it was thought that a tumor was essentially one homogeneous mass of cells, but more recently it has been shown that actually, in fact, there are multiple different cell populations within a single tumor. Now, why is this important? Different cell populations will behave differently, and there will be they will behave differently when it comes to therapy. So some might be sensitive to a therapy and the tumor will shrink, but there might be cells that are resistant as well. So trying to understand the heterogeneity and understand the different cell populations within a tumor is crucial in finding the best treatment strategy for each individual patient. So this is where TRACERx comes in. TRACERx is a national prospective study looking at early stage non-small cell lung cancer. And the aim is to recruit 842 patients with early stage non-small cell lung cancer and follow them over 5 to 10 years, trying to understand and get a better grasp of intratumor heterogeneity, the effect of that on treatment, as well as any metastases and metastatic process as well. So the aim of TRACERx is, like I said, to recruit early stage non-small cell lung cancer patients. In this case, the primary treatment is surgery with curative intent. And so during surgery, the entire tumor is taken out from the patient's body. And we can then perform multi-region sampling, meaning we don't just take a single biopsy of a tumor and only get a small subset of the cells that are actually present, but rather try and capture as much of the heterogeneity as possible by sampling as many regions as we can. And so for each of these different regions, we can then perform various analyses to detect the mutational information and also copy number data, and to try and basically classify these across the different regions. And so the idea is to start looking at mutations that are specific to the tumor that are acquired during the evolution of the tumor, and to use those to essentially time certain events. So here we use one quite important assumption, which is the infinite sites assumption, that says that mutations are rare events, and the genome is quite large. So the probability of hitting the exact same location across the genome multiple times is basically 0. And so with that, if we see the same mutation occurring in different regions of the tumor, this must have been a shared event that occurred early on during tumor evolution. So using this kind of assumption and this hypothesis, we can start to reconstruct some of the tumor evolution just by sampling at one time point, but using multiple samples. And so the idea is that we can time events, so mutations that occur in all of the regions of the tumor must have occurred early and were found on the trunk of the tree, whereas mutations that occurred later during the tumor development and only occur in a small number of regions have occurred later in tumor evolution, and we call those subclonal mutations, or they occur on the branches. And so now to just kind of go into the a bit more detail of the phylogenetic reconstruction that we do. So like I said, we mainly use mutations because there we can use them as essentially a way to time events. And so we can look at all the different mutations that we find across all of the different regions. And we can look at presence or absence of the mutations in each region, and also at the cellular prevalence. So how many of the cells do we actually find this mutation in? So in some cases, we might find it in all of the cells within the region, but in some cases, we might only find it in 50% or 20% of the cells. And using this information, we can apply some clustering algorithms that basically give us a approximation of the subclones present within the primary tumor. So this is kind of thinking back to the intratumor heterogeneity. We can use these subclones to essentially infer the different cell populations that we find within the tumor. And then we can look specifically at each of the regions and see which subclones are present and at what proportion, and also essentially reconstruct a tree using the same approach. So what we end up with is a tree that was constructed based on the mutational profile of the tumor using the multi-region data that we have through TRACERx. And so each of these clones along the tree is essentially a cluster of mutations that appeared at a similar point in time. And so in the first instance, what we've done is we've used all of the mutations that are present to now perform inferences, but we do also have additional information about some of these mutations, and which we can then also map back to this tree as well. And with that, I'm going to hand over to Cris.
Yeah, thank you, Ariana. Yes, so this slide is basically a representation of the actual data that we have. So the first line basically represents one single tree, and in particular represents a tree with 3 branches. So all the 3 plots on the top are all part of the same patient. This represents basically the cancer evolution of one single patient. And basically this is a representation of the tree that has been obtained with the technology that Ariana's group TRACERx has developed and that she described. The same reasoning can be done for the bottom image, the 4 plots in the bottom of the slide. which in this case it represents another tree, so another patient. In this case, it's a tree with 4 branches. So the number of branches across trees vary. Also the number of time steps across the same patient, so vary as well. The difference between the first and the second is quite important, and is basically the sort of first challenge that we have, quite a complicated one, and it would be absolutely a winning situation to be able to, you know, solve this challenge, is to understand whether the cancer corresponding to each of these 3 is going to reoccur, is going to happen again in the future of the patient. Remember that these are all tumor masses that are quite big in size in some sense, and have been extracted from a patient. And so a natural question is, will this cancer come back eventually? A more refined question, which is in our to-do list, would be in how long? If yes, in how long is the cancer going to come back? Yes, so if I can have control, thank you. So yes, so now going back to signatures and rough paths. So what we're dealing with is a very, very similar, which is almost, you know, this is all one of the motivations why we wanted to initiate this collaboration, which came chronologically after the one with Tom and the defense and security, is the surprisingly similarity between the data that is involved in cancer research, like this one, and, for example, malware detection, like the previous talk. And basically, the reasoning that we do on these sort of objects, which, as you can see on the left-hand side of this slide, are what we call streaming trees. Basically, they are tree-like structures where the information from one node to the next branching is sequential. So all sorts of things can happen between the current node up until the next branching. And we, with path signatures, we can capture this complex sequential information, and we can actually disentangle these trees in a very simple way by simply considering each single branch. So if you start from the root node and you go up until one of the final nodes, and you do that for all final nodes, well, you basically have disentangled the trees into its component, into its branches. And what are branches? The way we see branches is ensemble of paths. The collection of all the branches of one single tree is one single cloud or ensemble of paths with, in the specific case of trees, common history. So the different branches is not very well represented on the right-hand side here, but basically the different branches are multivariate time series with some sort of superposition in time up until a certain point in time where they start to differ. But they're still a single ensemble of single cloud. Right. So what are the challenges for this specific project? Well, first of all, we have 91 data points. So we have 91 trees, 91 patients. So obviously, you know, deep learning is a very tempting option for inference, but in this specific circumstance, it cannot be possibly deployed, because with 91 samples, there is nothing much you can do with neural nets. The second challenge is the huge dimensionality of these channels. So signatures and rough paths and in machine learning have been traditionally used to consume and learn and do inference on moderately low dimensional streams. And by dimensional, I mean the number of channels in your multivariate time series. And this is because, as Terry was explaining, the amount of information that is contained in a sequence of events is exponential because of the order of events. The non-exchangeability of the timing points of a sequence is what by nature creates this exponential explosion in the amount of information. So exponential in what? Exponential in the number of channels. So if you start with the time series with 3 channels, well, you will get 3 features at order 1 signature, 3 squared order 3 squared, so 9 at the level 2, 3 cubed 27 at level 3, and so on and so forth. If you have 16,000 channels, like we have in this case, because we are actually plus 60,000 plus. We are monitoring the amount of, well, the accumulative number of mutations appearing in each single gene that we are considering. So there are more than 16,000 genes that we are considering and look at their evolution over time. It becomes quite, I mean, almost, you know, 16,000 squared. That's just signature level 2. It's already something that is quite a huge number. So it's you know, it's a challenge. It's a challenge for signatures to deal with this sort of very, very high dimensional streams. And then there is also the heterogeneity of the data itself. Heterogeneity in the sense that within the same patient, for example, there can be a different amount of branches. So one patient can have 3, one patient can have 4, like we have said before. And the number of time steps that are basically given to you by the TRACERx tree algorithm can differ as well across different. So the length of the time series, or the depth of the trees in this case, can be different across patients. So the goal basically is similar, in line with what Maud and Tom presented before. It's more generally is the one of trying to build a sort of way of measuring the similarity of 2 non-standard objects, like 2 clouds of paths, 2 trees, like the ones that we've seen so far. So the scope is to find a way of measuring the similarity between 2 such objects. Why measuring the similarity and rather than extracting features? Well, extracting features and measuring similarity are 2 machine learning, almost orthogonal machine learning ways of doing inference. And the extraction of information in order to learn a function is very much what the signature does, which, as we saw, as I've said, it's almost impossible to do in this case because of the exponential explosion in features. Well, what we do in this it's that instead of extracting features and learn functions, we can measure similarity between pairs of objects and use kernel methods and use support vector machine, use Gaussian processes, and so on and so forth, provided we have a kernel on such objects. Now, Maud, in her talk, she explained how starting from the signature kernel, which is basically a kernel on time series, one can construct the distribution regression kernel, which is basically a kernel on clouds of time series. So what that kernel allows you to do is to measure how similar a cloud of an ensemble of time series is compared to another ensemble of time series. And trees, as I said before, can be seen as ensemble of time series, and therefore it's a simple consequence to basically use this distribution regression kernel to measure the similarity between 2 trees. Now, the advantage of one of the key advantages of the signature kernel is that contrary to the signature that explodes whose dimension explodes with the number of features, it doesn't suffer at all from that explosion in dimensionality. Its computational cost, thanks to the kernel trick that comes from different ways of, in this case, we use the PDE solver to compute this kernel. The computational cost for computing the kernel is linear in the number of channels. So the inference on paths, on time series of huge dimensionality becomes feasible. It's quadratic in the length, which is a bit problematic for long time series. But if you have long time series and a very high dimensional time series, then I don't know, you're just trying to capture the whole universe in some sense. But you know, that's challenge by challenge in some sense. Well, anyway, so we can, as I said, we can compute the distance or a kernel between two trees, and we can do so for all pairwise or pairs of trees in our data set. And we form this matrix on the right-hand side, which is called in the community is called the Gram matrix, and the ijth entries of this matrix is simply the kernel between 3i and 3j. And once you have computed this matrix, which is a compact object, it's a 91 by 91 matrix, well, then you can feed this matrix into your favorite kernel method, Gaussian process or support vector machine classifiers, and perform and carry out basically the final inference. So yes, so this is basically where we're at. There are future challenges we're trying to little by little explore and unveil, which is quite exciting. And you can find this the code for the signature kernel, which should be easy. It should be easy to use for everybody who's interested in this GitHub. It's the result of many collaborations. It's Maud, for example, she implemented the GPU part of the signature kernel. James Foster, I don't know if he's connected, he found a very performant PD solver to solve this specific Gursa PD that appears in the signature kernel. So this is a very nice, I think, result of the intra-group collaboration of DataSig. Yes, and that's the end of my talk.
Thank you very much, Cris and Ariana as well. I see that there are a number of questions already, so let's work through them. One of them was posted in the chat, which was, have you looked into transfer learning and using at least maybe inner layers from pre-trained models?
I cannot see the question.
That one was, I think that one was in the chat rather than the Q&A.
Oh, okay, sorry. Yes. Have you looked into transferred learning? There is no neural net here, so I don't know what is meant by transfer learning. Transfer, as far as I understand, transfer learning is that you pre-train your parametric model on some task, and then you change the data set and you see if you transfer over the performance on another data set. In this case, there is no, I'm not sure. We have simply operated on a train test evaluation procedure where we have trained the SVM. In this case, we've used on the training set and tested it on the test set, and then seen the performance on the. And you see that this splitting into train, test, and evaluation is already problematic because we have 91 patients.
Okay, so another question in the Q&A now. Maybe try something like unsupervised training on deep learning. Models like transformer are expressive in many cases.
Absolutely, I agree. Transformers are the state of the art, but transformers with 91 input-output pairs, it's just not feasible. So in this case, what the challenge is really the small data regime. Yeah.
And one more question. Have you tried, or are you planning to integrate these findings with targeted therapies to study relationships between the tumor evolution and therapy resistance?
Maybe Ariana, you.
Yeah, so I don't know if it's possible to really integrate kind of targeted therapies directly, but one of the things that we are looking into is at the moment we're just using all mutations and all genes. So the idea is to maybe limit that down to specific mutations, I guess like mentioned here, driver mutations in known cancer genes like EGFR, like P53, etc., and kind of focus specifically on those. I guess, yeah, integrating the targeted therapies would be interesting, but I don't know if it's something that we can really do with kind of the data that we have. But it is definitely interesting to consider.
Okay, Ariana, Cris, that was great. Thank you very much. And to the previous two speakers in this session as well. We're going to take a break now for lunch, I think, which